Hello, and thank you for joining us for the sixth annual Global Summit on Kidney Disease Innovations. Vox Populi, time is now to prevent and cure kidney diseases. The summit is hosted by American Association of Kidney Patients in partnership with our allies from the George Washington University School of Medicine and Health Sciences. My name is Vasundhara Raghavan, and I'm an AAKP International Ambassador from India. I'm also a living kidney donor. I represent Kidney Warriors Foundation that mentors patients for better health and renal diet. Several books on real life instances of people with the disease have been published in the past. I'm a member of the AAKP since 2020. It allowed me to participate in actively in summits and gave me speaking opportunities that has helped me to do my work in India. It gave me an understanding of AAKP's policy making initiatives, and I recognize, as does the world, that AAKP has been the leadership in this area. This year's summit, Vox Populi, was purposely and strategically chosen by AAKP for its historical meaning, voice of the people. I'm one of the people who has been chosen to raise my voice in support of greater care, choice, and innovation in kidney care. And I'm one of the voices demanding a change in the status quo kidney medicine. Over 850 million people across the world have kidney disease. The number is increasing at an estimated rate of 5 to 7% annually. Every year, we lose thousands of innocent lives to preventable kidney issues and the lack of access to treatment. AAKP believes kidney disease is both a workforce and healthcare issue. The disease takes a devastating toll on human lives and livelihood. Most people understand that kidney disease, left undetected and untreated, can lead to premature death. But many are not aware that it impacts a person's dignity and ability to provide for themselves and their families. Kidney disease and outdated treatments can lead to unemployment, underemployment, disability, and dependency. In 2019, in 2019, AAKP declared the next 10 years the decade of the kidney. The time is now for kidney patient consumers, healthcare professionals, researchers, industry, and all the people of goodwill to unite our voices and demand change. We are optimistic we can and will save more lives. But to do this, we must demand new screening guidelines and diagnostics, new biologics, and new devices. Together, we can manage and solve this growing global public health crisis. The time is now for elected and appointed leaders worldwide to listen to patient voices. Governments must remove the regulatory and payment barriers they create that delay innovation and prevent timely care access. If government fails to act, we must raise our voices and demand an end to negative government detriments of health. Make no mistake, every nation is impacted by kidney disease and millions are at risk. The optimistic news is that every person watching this program today can help those impacted by disease and make a difference within their own nation. Through AAKP Global, AAKP is educating patients, care partners, healthcare professionals, and policy leaders across the world on unmet patient needs. Our annual Global Summit on Kidney Disease Innovations has helped put a human face on what is now the 10th leading cause of death based on data from the World Health Organization. We have connected people across the globe in a common purpose larger than one person or nation. Together, we have increased hope and optimism. So, thank you for joining us. Your voice is the voice of the people, and you can count on AAKP to keep amplifying throughout the world to create change. With that, I'm honored to serve as a moderator for our session entitled Alternatives to human organs, artificial, implantable, artificial wearable, and 3D printed kidneys. Prepare yourselves for an extraordinary session. Today, we are privileged to hear from remarkable individuals who are revolutionizing the field of transplantation. You will be inspired by the pioneering work that is happening. It is with great pleasure that I introduce our next speaker, Dr. Buddy Ratner. Dr. Ratner is the co-director of the Center for Dialysis Innovation, the director of the Engineered Biomaterials Engineering Research Center, the Michael L. and Myrna Darland Endowed Chair in Technology, Commercialization, and a professor in bioengineering and chemical engineering at the University of Washington. Today, he joins us to share the latest advancement in the Center for Dialysis Innovation's ambulatory kidney to increase vitality innovation and artificial kidney system. Dr. Ratner co-directs the Center for Dialysis Innovation with another esteemed ally of AAKP's, Dr. Jonathan Himmelfarb. Under their leadership, their research team has received 3 KidneyX awards for their groundbreaking work. Ladies and gentlemen, I turn the presentation over to Dr. Ratner.
Pleasure to be able to present a talk to you. I'm going to thank our host, Vasundhara, and the AAKP and George Washington University for this wonderful global summit. I'm going to talk about, you see the title there, Taking Kidney Dialysis into the 21st Century. And I think we have a ways to go. So what is this talk all about? Well, kidney dialysis has a history, certainly going back to the 1960s. I'll actually show you a little bit earlier than that. I am hardly saying anything new when I say it's far from optimal for the patient. We're going to talk about ways to make this much more patient-centric. What are the barriers holding back innovation? I'll go over some of those. Ultimately, I'll be telling you how the University of Washington and a startup company that I am a founder and stockholder of, Kuliana Technology, will transform how we do kidney dialysis. So again, to sort of set the stage, every major city has multiple dialysis centers. Most small cities have at least one. These dialysis centers sustain the lives of 4 to 5 million people worldwide. So maybe you think that dialysis is a solved problem with so many dialysis centers, an old-fashioned theme. I'm going to argue it's hardly a solved problem. So we're going to start here in, as I said, a bit of a historical introduction. The image on the left is Dr. Wilhelm Kolff. And Dr. Kolff, as a young physician in Nazi-occupied Holland, was concerned that he could do nothing for patients who showed end-stage kidney failure. And he thought about the idea, had some ideas, how one might remove urea and other toxins from the blood of a patient whose kidneys had failed, and built under very primitive conditions, very crude dialysis machine from a Ford water pump, washing machine parts, orange juice cans, sausage casings. Built the machine you see on the right. The woman in the little image at the bottom there, Sophie Schaffstadt, was the first human in all of history to have been brought out of a uremic coma. When the uremic toxins build too high, you go into a coma, and that has always led to death. She's probably the first human on this planet to have come out of that uremic coma on Dr. Kolff's machine. The story Dr. Kolff says is when she opened her eyes, is the first woman in history to be restored with such a machine. When she opened her eyes, her first words out of her mouth was, "I'm going to divorce my husband." It's a kind of entertaining story, but in any event, let's go on the history in the next slide. We're going to kind of shoot a little bit ahead to the 1950s and 60s. Dwight Eisenhower was president. Elvis Presley was popular, and if you were lucky, you had a 1955 Chevy convertible. And the key themes that we found in the sort of biomedical engineering literature at that time are the kidney, the heart valves, blood vessels. The field was just starting then. We're going to talk about the artificial kidney. We're up to 1960 here, and this is at the University of Washington, my home institution. And we see in this image three gentlemen. In the center is Belding Scribner, MD nephrologist, who was familiar with Kolf's work, but because of the nature of that procedure, couldn't do chronic dialysis. They could do acute dialysis, a few dialysis sessions. And for patients that had permanent end-stage liver failure, they still would succumb after a few treatments. Dr. Scribner was concerned. To the left is Les Babb is what we called him. I knew him well. And Dr. Babb was a chemical engineer, and to the right is Wayne Quinton, biomedical engineer. This collaboration between physicians and engineers led to a therapy that has led to sustaining the life of almost 5 million people each year now, namely dialysis treatment as we know it. So what Wayne Quinton, working with Belding Scribner, did was invent this access device so it didn't require a surgery every time to get access to the patient's blood. They could sort of plug the patient in and out. The famous Quinton Scribner shunt had a lot of complications, but still set the stage, revolutionized the field of dialysis as we know it today. That device was used on this man, Clyde Shields, the first individual who went on this chronic dialysis, which resembles very much what we do today. The apparatus obviously is very different looking, but the basic procedure was the same. Dr. Scribner appreciated that patients could not be coming into the hospital 3 times a week and felt that the idea of a dialysis center was suitable. And in fact, in Seattle, the world's first dialysis center was opened, led by Dr. Belding Scribner in 1962. Well, here we have dialysis centers. We have similar dialysis treatment. 60 years have passed since these pioneering efforts. Well, one of the other things that happened is I started my research work, my PhD work, and I did my thesis on actually urea removal and new kinds of membranes. So I've been involved in this type of work for quite a period of time. And here we are, let's say 2000 plus. We have patients using a system, a dialysis machine that looks pretty similar to what we're doing today. You notice it's pretty expensive. In the first decade of the 21st century, we spent 2000 to maybe 2010. We already spent greater than $2 trillion on dialysis, and I'm sure that's continually increased. So what are the problems with kidney dialysis? Well, there are access and disparity issues. It's complicated. It's not available in all places. There's a large number of individuals that simply don't have access to this, and the outcomes are sad. It's very expensive. It's environmentally unsound. And then there are technology medicine issues, poor outcomes for patients, poor patient satisfaction. And that's mostly what we're going to develop, mostly what we'll address here in this talk. So just to talk about the environmental impact, we use an incredible amount of water. We use about 400 liters of good quality water per treatment. And you multiply that out by people going 3 times a week to dialysis centers and maybe 4 million people worldwide, and you get 129 billion liters of water. That's 120,000 Olympic swimming pools of water, 120,000 that we dump down the drain. Also uses a lot of electricity and a lot of waste produced. So this is just one example. Just the PVC tubing sets we use in dialysis would go to the moon and back 3.5 times, and all that tubing is just chucked into the waste. One of our thinkings here was your kidneys work 24/7, and how can we expect 3 4-hour dialysis treatments a week to emulate the natural kidney? And kidneys are very clever organs. They do all sorts of things besides just getting rid of urea. They do remove wastes, but they keep the electrolytes in balance, pH control, water balance, produce hormones. They do all kinds of good things. So it's very hard to emulate a kidney with a machine, but let's see how much of that we can do. One of the thinkings, of course, was when we first started our effort here at the University of Washington, was maybe we could use modern tissue engineering technology to grow a new kidney. And there's huge potential in that, but it's really been 35 years since the inception of these ideas of tissue engineering. In 35 years, actually, the tissue engineers have done some remarkable things, but it's actually had almost no negligible contribution to sort of modern medicine, what's going on in medicine today. So we figured, hey, we might be able to do this, but it might take a long, long time to get to patients. We concluded as sort of a mechanical biomaterial strategy will be faster to get to patients. So let's talk about a wearable artificial kidney. Kind of our predicate for this was, if you look at the artificial heart or the heart assist device, in 1993 it was a big clunky machine. By 2007 it had been reduced to rollerboard size. And today it is you can hook one on your belt. And this gentleman all the way on the right there is having his heart supplemented, supported by an artificial heart while he's enjoying life under battery power. That's kind of our predicate or our model for what we'd like to do with kidneys. And there's a lot of history there. People have been thinking about how you might do this, starting in even 1965. But you'll notice we don't have any wearable or portable artificial kidneys today. Wearable kidney dialysis, really one of the first modern attempts was by these three gentlemen. In the center is Dr. Victor Gura, who's an inventor and invented a wearable kidney. To the left is Professor Jonathan Himmelfarb. Jonathan, as you'll see, became my colleague in developing new types of portable and wearable kidneys. And to the right is Professor Larry Kessler, who's an expert on FDA regulation. And we had a clinical trial of Dr. Gura's wearable kidney here at the University of Washington. You can see this young lady to the right is just absolutely thrilled to have this portable device compared to the large bedside equipment. The only problem with this was that the clinical trial was cut short. It was supposed to be up to 24 hours. It's cut short after a few hours because of concerns for patient safety. It was a brilliant idea, but maybe before its time and really was not working well. And so the clinical trial was cut short. That was the inspiration for allowing Dr. Himmelfarb and myself to launch at the University of Washington the Center for Dialysis Innovation in 2017. From day one, brought in patients. And one of our patients said, this is ridiculous. We need to find a better way to do this. And that really became sort of our mantra, our motto. So this is the center leadership. Jonathan and myself were co-directors, but there's Dr. Kessler working on regulatory aspects, Cassandra Thompson working on IP, filing IP, Glenda Roberts, who heads our patient advisory board, Anna Galperin, who is our scientific manager for the program, and Barry Fulkerson, who is an engineer. So Barry was an industry leader, 35 years experience in the dialysis industry, really helped develop practically every dialysis machine on the market today. So Barry was a great addition to our team. For example, I know a lot about the technology and how to develop new technology. What Barry knew is how to build a machine and how to bring that technology into a machine, a very important part of our partnership. Again, we have our important patient advisory board, chaired by Glenda Roberts, and we tried to bring in feedback from our advisory board consistently so we knew that what we would develop, maybe patients would embrace it, and that would lead to its success. Our vision was what we call the active, the ambulatory kidney to improve dialysis. Really, this illustration was quite, we'd call it just aspirational. It's just an illustration, but maybe it's where we're hoping this whole project might go. More realistically, we realized maybe a first generation might be a portable or roll-along device, and then ultimately, maybe into the future, a wearable. But what do we need to do such a device? Well, it has to be portable or wearable, miniaturized, water efficient. It should be environmentally friendly. The patient on maybe longer or continuous dialysis should have minimal anticoagulation for blood clotting. It should be robust, cost effective, patient centered, and ultimately restore kidney health. So, what is needed for a portable or wearable kidney? Well, I'm not going to go through this whole list. It'll take a while to read it. But we got to start with removing about 15 grams of urea a day and other toxins. And then also, if this is going to be light and portable, we want to work with 1 or 2 kilograms, liters of dialysate solution. No hookup to a water supply. And then a bunch of other criteria. So again, to emphasize this point, take about 400 liters of water, put it through that water system on the bottom. It gives us the 120 kilograms, 120 liters of good quality water that is used to dialyze the patient. What we want to do is do it with just 2 kilograms of water rather than 120. To make that all succeed for a portable or wearable, there are a lot of, let's call components, and they all have to fit together. That's the engineering part. What I have them is sort of outlined here, and I'm going to go through a few of these. I don't have time to do them all, but talk about some of the challenges and some of the ways we are addressing each of these pieces of the puzzle that must fit together to make this portable or wearable dialysis, the active dialysis, successful. So let's examine some pieces of the puzzle. So what are the challenges? Again, removing toxins, we have to remove about 15 grams of urea a day, do it with 1 or 2 kilograms of water, address other toxins, measure and analyze the toxins, and address electrolyte balance. And we have a number of investigators who have been working on this. This is the solution we came up with. It's been described in the scientific literature. It was a great scientific advance. And to the left in this illustration, you see a dialyzer. The patient would be at the far left, blood going through the dialyzer. And this is the dialysate loop. The dialysate goes into that device to the right, which has UV lights. And what we're doing is photocatalyzing, using the UV light, catalyze the breakdown of urea to nitrogen and CO2, two harmless gases that go into the atmosphere. Then the regenerated dialysate goes back in through the loop. So basically, we don't build up urea in the dialysate. We don't have to dump the dialysate. We can keep on using it over and over again. So does it work? Well, we built kind of a model. We used a 40-liter tank. Your body has about 40 liters of water in it. And we said, let's flow into that tank about 15 grams of urea per day at about the rate you make urea. And so if we're just flowing in urea into that tank, the urea level is going to build up. If you look at the little graph on the left, red line shows the increase in urea concentration over time because we're flowing it into the tank. But then we turn on our, what we call it, our dialysis regeneration module, those UV lights in the catalyst, and now the urea remains constant. Why? We're taking the urea that goes into that tank and we are converting it to CO2 and N2, and it's gone. The graph at the right also shows the dialysis regeneration module can break down most of the kidney toxins, other toxins that are made. So it is very effective in getting rid of these components. One of the other, let's call it, benefits of this system is that the UV light make the dialysis loop self-sterilizing. So what we're showing here is some experiments where we fed large concentrations of Staphylococcus aureus into the dialysis system. The orange is when we turn on the pore lights, or the photooxidation urea lights, the UV, and you notice the Staph aureus concentration goes down to zero very quickly. So self-sterilizing is another benefit of these UV light systems. We also looked at the, well, again, we're concerned with bacteria and biofilms. It's a big problem for dialysis patients. And another part of what we're doing is coating many of the components within our new dialysis system with a coating called carboxybetane, let's call it CB for short. And what we have here is fluorescently labeled bacteria. And again, this is a different bacteria, but also a big problem for dialysis systems. We have two kinds of bacteria here leading to sepsis, patient infection. And the uncoated system, you can see, build up large amounts of these bacteria because they're fluorescently labeled. When we coat our surfaces with the CB coating, we have almost no bacteria on. So it prevents the bacteria from sticking and forming these biofilms, which is very important. The CB coating is also extremely effective against blood clots, so it's making the surfaces more blood compatible. Well, let's just talk quickly about dialysis access. There are three ways we do this. The fistula, for years we said fistula first. I think now the medical community is appreciating that the fistula can be harmful and may not be the most optimal way to get patient access. Plus, it's very damaging to the arms. Another way is a vascular graft. Vascular grafts have a big problem of failing very quickly, and so they haven't been recommended. And then there's a central venous catheter that has a lot of issues with infection and clotting. So every one of the ways we do dialysis access has problems. And our center, University of Washington team, is looking for ways to get much better dialysis access. What we're doing is doing better vascular grafts and also improved catheters. So blood access challenges, I think I mentioned many of the challenges. The vascular access grafts fail 50% in 1 year. Central venous catheters fail, thrombosis and infection. The skin interface is a major challenge. And how can patients interface their bloodstream without these repeated sticks with big needles? So here's some new ways to think about the blood access, two approaches. We're making a more reliable, long-lasting vascular grafts. It will still involve the needle sticks, but if it does not fail at 1 year, the patient's lifeline to dialysis is still maintained. But ultimately, we're developing a central venous catheter that is both safe and needle-free, and we call this the active connection. Better vascular grafts. We have a rationale for developing a vascular graft. Why do they fail? We think vascular grafts fail due to inflammation. The body literally attacks them, never stops attacking them. So we're addressing inflammation. So this is our reimagining, rethinking of vascular grafts. We've invented a material with a unique porosity, unique holes. Each of those little spheres off to the left there are about half the diameter of a human hair, very small. On the bottom, you see a tube made of this spherical porous material. And in the upper right, what we expect to happen, we expect blood vessels, once implanted, blood vessels will ingrow from the tissue and literally reseed the luminal surface, the inside of the blood vessel, with living endothelium, just what your blood vessels have. In other words, we're sort of recreating, re-engineering your own blood vessel in our synthetic or dialysis access vascular graft. One of the things we want to do in this vascular graft is have the same mechanics, same flexibility as native artery. And so we've engineered some materials so we can achieve the same mechanics, the same flexibility as native artery. It feels like a native artery. So we invented these grafts, and working with Dr. Lina Quiroga, who's a vascular access surgeon, she does humans every day, we put them into sheep. Sheep is a good animal model because they heal slowly, like humans do. And so if you look on the right, there's the conventional Teflon vascular access graft. And when we opened the sheep up to examine what was happening, Dr. Quiroga is busy pinching on this dense fibrous capsule that surrounds the graft. This is the attack the body makes on the graft. When she opened up on the left, what we call our 6S graft, she just saw what looked like a living beating artery. And her own words was, "This is a surprise." She said, "I knew I surgically inserted a vascular graft, but all I see is a living beating artery." Well, that was exactly what we had hoped to achieve. When we examined these grafts. One of the important aspects of, again, a living artery is this cell coverage on the lumen or the inside surface of the tube, living cellular layer, much like our natural blood vessel. And you can see our 6S graft to the left has much higher than the existing Teflon graft. This is just more details on that, what we call luminal endothelialization, this rehealing. But the 6S or polyurethane graft, which is the bars on the right, you can see the total is much greater than the Teflon, which is the black bar, let's say, on the left. We're well on our way to hopefully bringing that vascular graft to first in human. We're working with the FDA on how to do that. But we're also working on, within our program, on what we call the active connection. And that device shown in sort of a mock-up or scale model on the right would be just a plug-in, plug-out that would be connected to a central venous catheter. There'd be no more needles. It's needleless, touchless, self-sterilizing, has built-in sensors to understand what's happening. It heals into the skin, so the skin infection is eliminated. Resists clotting, prevents infection on the inside due to the CB coating, and we think this could be a total enabler for home dialysis. No more needle sticks. So we're very excited about this too. So a key challenge in this whole project is how to get all these innovations to patients. How do we get university or academic developments to patients? And as we say, publishing a paper in a high impact journal is simply not enough. So we've launched a company. Companies know how to get product to patients. Kuleana Technology. Kuleana is a Hawaiian word meaning responsibility and care. That's my company too. Again, we are involving the patient voice with patient panels, patient advisory board. The patients really have to tell us what they want. So in 1963, Dr. Belding Scribner, the great pioneer in our field, wrote, "If the treatment of chronic uremia cannot fully rehabilitate the patient, the treatment is inadequate." He wrote that in 63. We think it's still true, but we're hoping the work our team here at the University of Washington is doing will bring something closer to a rehabilitation, a normal life to kidney dialysis patients. I really have to give special thanks to my enthusiastic students and staff. Here you see some of them just celebrating, but I want to also thank you all for paying attention. Look forward to questions.
Thank you, Dr. Ratner, for this wonderful presentation. I do have a few questions to ask you. Dr. Ratner, this year's theme is Vox Populi. Can you tell us how important it is to you as a professional and leader in artificial organs to know the voice and demand for change support your work? Does it fuel determination to deliver a workable device in this decade?
Well, we are aiming to certainly do that. I would even be so optimistic as to think within the next 5 years we could be into early human trials on this. The work has progressed very well. We've been doing these developments for 7 years, starting from zero. And we have some systems that we believe are workable, and we are ready to translate them to patients. So yes, with the patient voice guiding the directions we're going in, we hope to bring this technology to patients very soon.
Dr. Ratner, can you tell our viewers how important the nexus is among universities like University of Washington, private sector investors and companies, and patient advocacy groups like AAKP when it comes to advancing artificial organs? How dependent are each sector upon one another?
Well, I think what we have here is sort of an ecosystem. Each of them requires the others. Again, the major elements nicely summarized there Our universities are very good at sort of basic science and basic research. Companies are very good in taking that basic research and converting it to product that directly benefits patients. And then finally, the patients, AAKP, and other advocacy groups can bring this, can ensure that it meets the needs of the patient and gets to the patients. So I see this as a teamwork. Again, an ecosystem, I think, is a good word. All the components have to fit together or we're out of kilter.
Dr. Ratner, in 2019, AAKP called the coming decade the decade of the kidney. Can you tell us, as a medical professional and innovative leader, are you confident we will see clinical trials for artificial organ technologies during this decade?
Yes, I am confident. I'm quite confident of that. Our group here at the University of Washington has made great progress, but a number of other groups worldwide have also developed important, unique approaches to many of the problems I think I've outlined in my talk. And I think this will be a very important decade, let's say 2020 to 2030, where we will see a transformation in our ability to improve the quality of life, improve outcomes for end-stage kidney failure patients. And I think we're on path to see some exciting changes in medicine that we have not seen since the 1960 development of Dr. Belding Scribner.
Again, Dr. Ratner, we appreciate you being with us for AAKP Global Summit on Kidney Disease Innovations and for your team's commitment to change status quo kidney care by innovating beyond dialysis.